So I think we can start. It's a pleasure for me, but I think for all of us to have here Professor Jan Pawlowski. He's from the Department of Genetics and Evolution at the University of Geneva in Switzerland, where he leads the research group on molecular systematics and environmental genomics. His main interest, he started his career working on the evolutionary history of foraminifera, combining molecular, morphological, and fossil data. And then he moved to molecular approaches, mostly on barcoding, to assess let's say, the environmental diversity based on environmental DNA barcoding, DNA-based identification of species, and the development of DNA tests for the assessment and monitoring of aquatic ecosystem. He is now involved in the DNA barcoding of foraminifera, the Foreign Barcoding Project, the genetic inventory of the Swiss biodiversity, and a few years ago he started a startup that is called IDGene that deals with the environmental DNA testing. And during his career he also organized a scientific mission in Antarctica, the Svalbard Island, as well as in Japan. Today he's going to talk us about how on the next generation biomonitoring, or how DNA metabarcoding can be used to assess the impacts on the marine environment. So thank you very much again, and we look forward to hearing your seminar.
Hello, and thank you for this very nice introduction and inviting me here. In this introduction you could see that I have been doing so many things. You may wonder what I am supposed to tell you. So I think I will say what I want to tell you is just a very simple message that you can go from very academic research to very applied research, and it makes sense. And I think that this is mainly what I will try to show you in these few slides about how to use this metabarcoding in applied work. So I saw here many people with whom I have been doing biomarks, and I've been here a few years ago, and we, as far as I remember, our main research objective was to explore the diversity of organisms and plankton, and also a little bit of benthos, and this was what we were supposed to do in biomarks. But in the biomarks, when you look on the proposal, there was also a paragraph, a point, about application of this metabarcoding data to something, much more practical. And I think this point never been really developed within the biomarks, and I was hoping that we will do biomarks too, but we never did biomarks too. And now I tried to do it on my own way, and this is what I will try to show you. So there will be a little bit of introduction about this topic, and then coming back to the, I think it's important to see what is bioindication in general for those who are not really ecologists or who has no real background on this field. So this is how you can assess. You can assess the environment either by physical or chemical parameters, which is very commonly used on the right side. And I don't know whether I can have any pointer here, or this. Yeah, yeah. So you can either use this chemical, or you can use the bioindicators. And this is also very common, that you use different group of organisms that can be diatoms or forams or macroalgae or macroinvertebrates that you can use as bioindicators. And these bioindicators would give you some kind of smiley depending on the work. So this is mainly done this way because politicians don't understand what this Latin names mean, so they just have to see whether a smile is good or not, red or blue, and this is a way that everybody's doing the same. Sometimes it's only three types of smileys, sometimes it's 5, but it doesn't change very much. So my idea that we could use potentially also DNA as another category of indicator, and this is what all this is about. So using DNA as another way to get to the bioindication. So this is one of the index that is commonly used for marine environment, which is AMBI. It's a ASTI marine biotic index that has been proposed, that is recognized by European community for all kind of benthos impact. And generally how this index works, so you divide the species into the category, ecological category, and then you calculate, it's a very, very simple formula. Depending on the group the species belong, then you give them special percentage of this species, and then you give a kind of ecological value to this person, to this group. So at the end, you have this formula of index, AMBI index, and when you look at this, you can, depending on the group, you can decide what the index is, and then you have to categorize index into the different groups, and then the different groups, different category will define which smiley you obtain. So this is a little bit the most simple way to translate the impact into some very easy politically, to things to understand. The formulas of most of indices is very simple. It has to have this, I would say, abundance, usually relative abundance of species. You have to identify species, and you have to assign species with some kind of ecological category. All indices work basically on the same way, except the very simple indices that are only based on diversity or richness. But more complex indices also use this category in the formula. So this is the way that you can evaluate the impact. And then, of course, the basic problem with this type of indices is that they depend on morphotaxonomy. You have to identify the species. So somehow, you cannot do the index if you're not good in morphotaxonomy, or at least trained taxonomists. And this is what happens in most of this agency or office that do this kind of assessment. There are people who go on the microscope and identify species. Whether they do it correctly or not, that's another question. But this is the basic on this. Because, in fact, what morphological identification is, it's only a step to get to the ecological group. Because otherwise, you could say that you cannot say what is the ecological group as long as you don't identify the species. Okay? You have the list of the species, you have the groups to which the species belong, and this is how AMBI or other indices works. You always have this list that assign the species to the ecological group. And our problem, our objective is whether we can do it differently. Whether we could use, instead of morphotaxonomic identification, whether we could use DNA as identifier. Can we use the DNA? Because we know now that we can obtain plenty of sequences using high throughput sequencing. So of course the obvious idea is why you could not use these sequences to obtain this identification, and then from there go to the index the same way. Does it work or not? And this is mostly what I will show you, and what the kind of issues and problems we have with this. Before, there's only a little bit introduction. I don't want to go to the details of molecular work. I suppose most of you know, but for those who are not really familiar with eDNA or metabar coding, this is just a reminder that eDNA, what is called unvolatile DNA or sedimentary DNA or water DNA, whatever the name is, correspond to all DNA that is found in the sample. And this DNA is not necessarily the DNA of organisms. It's also DNA that remain from the water or remain from the organelles. If you use, for example, on what we suppose now that there's a huge amount of what is called extracellular DNA. And this has been supposed to be just molecules of DNA. And we think that, of course, in the water there's a lot of molecules of DNA, but there's even more of organelles. In fact, a pint of pieces of cells or fragments of cells, and this is all combined, give you this total DNA. So somehow it's a problem. It can be a problem if you want to disentangle, for example, plankton from benthos, because of course, benthos sediments contain also all DNA that come from the water column. So somehow it can be a problem. It's not, very always obvious to know what to do with this organism. So this is one problem. Then, you know, I think this is for all molecular biologists is familiar with this workflow, though it's relatively simple. What we are doing usually is just to take sample, make DNA extraction. In some cases, we do also RNA, which make all this workflow much more complicated. And honestly, it's not possible to continue for routine work. So now we do everything on DNA. Then it's amplified, and then it's high throughput amplicon sequencing. So it's something very routinely done. We use Illumina for all the data, but can be done with any other type. And then, of course, much more complicated sometimes is this dry lab stuff, which is where you have to identify the sequences, you know, filter, cluster, taxonomically assign these sequences. This is where many problems appear. But generally, it's something that has been already very much developed by the people for academic research, so it's not necessarily very complicated neither. But it could be that many of these steps, or even these steps, at every step, in fact, you have biases, and you have to be aware about these biases to somehow make it, you know, applicable or standardized. Okay, and then out of this data, you can come to this formula of index. So this is the workload. And then, yeah, somehow we find out, when we started, it's relatively new, we started this work a few years ago, and we have been wondering how to really make it optimally. And this is the way that is done now. So you go on the microscope, you take the sample, you just assign this value to this from the table, and then you get to the index. Then the most simple way to do would be to do the same, but just get the sequences, assign the sequences. When you assign the sequences, you get to this morphological tables, you get the values, and then you get the index. But many of sequences remain unassigned or difficult to assign because reference database is difficult, and I will show you some challenges. So sometimes another way would be much more better to do something with this unassigned stuff, or what I will show you today also is a way that you don't assign at all, that you work with sequences directly without any kind of taxonomic assignment, which makes some problems, but it can work as well. So you have to be aware about this different way to process this meta-barcoding data. Okay, I will show you two projects that we are, I think, the most advanced. We have many different projects on, but these two projects are the most advanced, and most publication come from this. One is on the freshwater diatoms, benthic diatoms. So for those who are not freshwater, we are in the marine station. You have to be aware that there's benthic diatoms are used by regulators to assess freshwater rivers and streams in all European countries. That's an obligatory process. So it doesn't work for marine for some reasons. So then the other example that I will show you concerns the fish farming, aquaculture, which is also in some countries, especially Scandinavian countries, are very strongly regulated. And then we have been studied this on this case of salmon farms. And then at the end I also talk about the oil and gas industry, which is another topic. Okay, so the first case is diatoms. When you work with the morphology, what you do for these diatoms rivers, you just have to how you say, you take a brush and you have to brush the stones, and everything is very well standardized, so you have to use 3 stones from the middle of the river. You have to choose them. The stones usually are kind of have a biofilm on them, so you brush the stones and then put this biofilm into formalin, and then to process them and to look at the microscope. This is the traditional way to do with the diatoms. And then what we did for this study that we do in parallel when the people were taking samples for morphology, for microscopy. We took the samples for DNA. So the samples come, in fact, from exactly the same biofilm sample. That was brushed, the stones were brushed, and then were taken into, divided, split in two. A part was taken to the microscopy, and then the people were making the index out of this usual way. And this is the formula of the index. It's a little bit more complicated than the one that is used for the marine invertebrates. I will talk later about it, but there are 3 different variables for this index. But basically, it's just abundance, relative abundance, and 2 ecological values. And then we take the samples, do DNA from it, and then you also come to the index. So this was a study that has been already published in 2015. And basically, in this first study, which was like a pilot study, what we do, we just work on assigned sequences. So we assign sequences. Freshwater diatoms are relatively well known, so we could do it. And this was formidable, fantastic. So the results, when you look at this graph, you say it works. First time I've seen it was, wow, it's surprising. So what it to show you, these are these categories. These are the different colors correspond to these different smileys. So blue is a very unpolluted environment, then green, yellow, orange, and red is very polluted. I have to say that all come from urban area in Geneva. So as you could see, there's nothing really blue. On morphology, whatever. It's all polluted because it's all urban. But it's not necessarily very bad. The first column is morphology. Then the next one is RNA. And the third one is DNA. So we just do only assigned species. And we get to sometimes very good correlation between. They show the same. What we wanted is that they show the same category of pollution. We don't care about the names or whatsoever. We just wanted to have them on the same. So sometimes it works, sometimes much less. Like here, for example, at that time we use RNA. As I said, RNA get much better results when you look on the correlation. The problem is that RNA costs much more. So for the routine research, it is not possible to use. And sometimes it works, sometimes it's not so well. So we said, okay, we look what are the problems in this, where it doesn't work, okay? So here we identify several problems. First of all, when you do this type of assign approach, taxonomic assign approach, the first thing that the database is very incomplete. You cannot get more than half. Half is the maximum of sequences that you can identify. Half of them would just go unassigned. It's impossible to find out what it is. Then the other problem is that the same species, and then taxonomist diatoms shouldn't be surprised that the same species, morphospecies, of course, doesn't work anymore. Yeah, show many different genotypes. So this is what they called autus. So for the same morphotypes, you have several autus. Sometimes it's not so many, but sometimes it's very variable. So you have this problem of genotypes, different genotypes, and you also have this problem of non-assigned sequences. So somehow this problem consists in the fact that you produce this million of sequences and then you can only use part of it. You cannot use all of them. So of course this is not very good. And then, well, unfortunately you can't see very well, but this is what I told. This is about this problem of cryptic species. This is the case of coconaes. And coconaes have the ecological values. There are 2 species in freshwater that have different ecological values, but when you do molecular work, you have plenty of genotypes, and you don't know which value corresponds to which, because they has been decided on the basis of morphology. So you kind of complicate it, and you have to combine them into one kind of unit and forget about these ecological differences. So this is, of course, another problem, that when you interpret the genetic data, it's very difficult to really go to morphological data as per se. it's not directly related always. And unfortunately, it's impossible to do, except that for each of the scriptic species, you will have to give them the ecological value separately. And then the issue that is always the most critical is abundance. All formulas of indices include the abundance, relative abundance, but some kind of abundance. And of course, somehow you get quite relatively the same species among the most abundant species. It's here, and it's not very well visible, but the table often contains the same species, but never in the same position. And you can't really expect that you obtain exactly the same data. Even for diatoms. Diatoms are small, so you have a lot of diatoms in the sediment samples, but still, because of different reasons, probably different copy numbers, different way of amplification, some amplified easier, some amplified not as good, that you have all these biases. And this is one of the most critical points that people criticized, people like me, who trying to replace this indices, that we cannot get abundance. If you cannot get abundance, it's worth nothing, and you shouldn't use it. Because all ecologists like to count something. So if they don't count and they don't find the same numbers, they are upset. So this abundance bias is something crucial for the rest of the story. When you have these three challenges, reference database, cryptic species, abundance biases, then you understand that you have to find something else. That we are not happy with what we propose, and then we go to what we call taxonomy-free approach. So we decided, let's do differently. Instead of assign the species to some kind of reference database, we try to do it on the sequence directory. In this case, on the OTU. So you cluster your sequences into OTU, and then you provide ecological value to the OTU, whatever it means in taxonomy. It can be done only if you know, in the other way, you have to calibrate it. You have to know what is the ecological status of the place, of the site where you take the samples. Otherwise, it cannot be done. So to give OTU some ecological value, you compare where this OTU is in different samples, and then you find the majority of OTU is, for example, found in this particular ecological status, and this will be the value that is given to this OTU. Okay? And the result was spectacular. This is also published already last year. This is, in fact, this is correlation, but you can see that the points are much more closer. So the regression is much better on the right side. On the right side is taxonomy free. On the left side is normal assignment way. So you can already see that the correlation is better here than it's here. And also, each of this bar, this is about 40 sites, show the difference between molecular index and classical conventional morphological index. So the directive says that when you do the morphological index, you also have some kind of variability. So some people don't identify correctly, so it's accepted that this variability can be from 0.5 to 1. The scale here means the index goes from 0 to 8. So 1 is the difference that is acceptable, even for morphology. And this is what here you can see that here you have this threshold of 1, and all these samples are not conform to this threshold. It's above. So the difference was bigger. These are all these points that far away from the curve. And here you can see that this proportion is dropped down. It's much less of samples that makes a difference more than 1. So it's really why it's happened, because we use all data. And we use all OTUs instead of using only the assigned OTUs. This is the main reason why we were able to reduce this. So from practical reason, it's much better. Here it's explained, again, it's maybe not very well visible here, but it's explained the difference between these values. Because honestly, what is really crucial is this ecological value, because you need to give this value to make your index. So the value are assigned normally to the morphal species. Here we assign the values to the sequence, or to the author, in fact. In this show, unfortunately, the quality is not enough, but you can see this, if it's adjusted, you will see the presentation, that in many cases, unfortunately, you don't have the same value that you have for the assigned species. That's come probably from the fact that this is based only on a limited number of sites. So we still don't exactly have the same value, but you have a lot of sequences that you don't use as a normal. That's why the index was relatively good. And this is as another value. So in this Swiss index, we have values that is ecological values, which is just a way to say that this author or species is the most abundant in a particular ecological condition. And then you have this weighting factor that is kind of factors that take into consideration the occurrence of this particular species. It's because some species could occur very widely, it's very common, and then some other species occur in very few areas. So this is kind of ponderated, weighted factor, and then H in the formula is the relative abundance. So there are three, but in many indices, they don't use this weighting factor. Swiss people use it, but many indices use just ecological value and relative abundance. This is basically what indices do. Okay, and then, and this show you also the most important things, in my opinion, is that the dark bars indicate the number of authors that are used if you assign. And the gray ones are the number of authors that you use when you work without assignment. Because you still have some authors that you cannot use, have no any values. But you can see the difference. So you have much more use of the data in terms of reads and in terms of author. So this is the authors, these are the reads. The reads use almost everything, because most of these authors that are not here is just rare authors that somehow has been filtered out and are not really important. But in terms of reads, it means that if I spend money to make 1 million of sequences, I will use this 1 million of sequences instead of half of the million or even less. So this is very important, and this is why this taxonomy-free approach works. Okay, this is one part. Then another case study where we go even further. So this study changed totally, come to marine environment. And for those of marine ecologists probably know that one of the mainly used parameters is the benthic invertebrates. What is studied is the macroinvertebrates, benthos, zoobenthos, or things like that. So we wanted to have something very clear, because it's good for the pilot study to work on the case where you have clearly polluted place and clearly unpolluted place. Otherwise, it's complicated. So we choose these farms at the beginning mainly for this reason, because this is what they call point pollution. Farm is polluted, farm is sending all the food, and you can imagine each of the cage is contained from 30 to 50,000 salmons. The cage is often 20 meters deep, so it's a huge amount of fish. The fish, of course, produce a lot of feces. The food is coming, not food, all food is eaten by salmon, so food go down, and everything under the cages is theoretically polluted. And this is also regulated. There are regulations that say how much you can pollute. And that is what this pollution mainly is organic enrichment. That's what's happened in this case mainly. But we should not forget that there's also, in many cases, antibiotics, because fish sometimes are treated. And also the third problem is this anti-fouling treatment. So the cages are treated with copper or other things to make anti-foul, to reduce the number of organisms that grow on the cage, on the net. Otherwise, the organisms will grow totally the cage, and at the end, there will be no enough oxygen, no enough water flow, and the fish will die. So they treat these nets, and then, of course, this is another chemical problem. So there are many problems, but honestly, the main problem is organic enrichment, and this is what we mainly studied here. So actually, this is also a very practical problem, because in some countries, the regulations are very strict, and every year you have to assess your farm. This is the case in Scotland, Norway, or some other Faroe Islands and other countries, Canada. So what they do now, they take the samples, grab sample like that. The samples is sieved, and then the macrofauna is sorted. This is like a hamburger, you know, made from Capitella. I don't know whether Capitella is a kind of a polycats worm, and it's very often in the organic enrichment everywhere. So this is only Capitella. It's a very highly polluted place, of course. But of course, this is time-consuming. So someone has to pay, and there's a lot of every farm. It's 10 grams minimum. 10 grams have to be taken. It is 1 day of work on the ship, because every sieving and everything here is about half an hour or 1 hour of time, and of course you have to sort this. So we came there a few years ago to Scotland, and with my PhD student at the time, he said, okay, we can change it. We take 1 gram of sediment, we'll tell you everything what is here, and you don't have to worry. You don't have to sieve, you don't have to sort them, and so on. So they were very happy. You can imagine they believe it. Well, a little bit naively, maybe. But there's a real purpose on this, very practical reason to do it. So this is just to show you why all these problems that they have, you have to treat the samples, you have to, you know, analyze them on microscope. This takes sometimes more than 3 months. This is also another factor, how you can assess, you know, in the reasonable time if it's take more than 3 months. So, and the farmer has to they have to do it after the cycle, and the farmers have to have a good assessment before they put the new fish, and they don't want to wait 3 months. They want to put the fish immediately. So they wanted to reduce it, and then I must say they support our research very strongly to find out how to do it without all this, you know, without all this manual work. So, yeah. But also we get the project with a lot of research questions, like here, you know, the problem was we didn't know how this DNA really reflects the data in terms of macroinvertebrates. I have no idea, because this was the first time. It was not at all diatoms, not as microscopic organisms, the big worms, you know, echinoderms. So this is very important to know. The other question was, what if there are any factors that, you know, influences the analysis in terms of, you know, research on DNA, and then also, what about the biotic indices? Can we infer them from the DNA? So all this question we had to answer. And first, yeah, we did exactly the same, so I can go through. We did always morphotaxonomy and metabolic coding on all samples. So we always knew exactly what was the result from this classical approach, like in the diatoms. And then the first results was very positive. It's like with diatoms, we were very happy. In fact, what you see here, these are the result DNA, I think, or RNA. I don't remember which one is up. This is another molecular, DNA, RNA, and this is morphology. And without saying nothing, you can already say where the impact it is. Yeah? Where it is. Both morphology, DNA, RNA will show you the same story. You don't have to do indexes, you don't have to do nothing. You see exactly where the border of impact is. All this, you know, black, dark, red stuff is Capitella. When you go out of impact, Capitella disappeared. There's also some oligocats, tubifex kind of stuff, also here. And you see them on the morphology. When you go out of 60 meters, which is the regulatory region of impact, when you go 200 meters, you get this very different image. So DNA, this was for me, it's the most convincing evidence that when you do metabolic coding, you obtain very clearly the situation with the macrofauna, because this is macrofauna. So don't ask me how it is possible, how we can get echinoderms DNA from here. Of course, not from echinoderms, because we only use 5 grams of sediment. This is all from the extracellular or organelle DNA, but it's there. It's enough to get every group of taxonomy groups, while here everything is dominated by capitalids or all kind of opportunistic species that live in the organic enrichment area. So we don't need the index, but of course we do the index, and the index, not surprisingly, works very well. Even on the science sequences, we have to understand that this is all just by sequencing, assign the sequences to macroinvertebrates, and then don't change anything. And this is the problem, that done like that, it works, it can work, but only 1% of sequences were used, which means I do 1 million of sequences. Well, basically we do 100,000 per sample, and from 100,000 sequences, I use only 1,000 sequences. So it's 99,000 to the garbage. And this is not possible for me. It's a waste of, it works for, I will tell you, it works in a very contrasted place. And this was very contrasted area. But when you go to the place where the things are not much less impacted, then it's much more complicated, because there's not enough of signal, and this 1% will not be enough. So somehow we have to go out of this problem. Here there's another slide that shows just for those who don't like abundance and criticize the abundance, you still have clearly Capitella close to the cage, fish cage, and very few Capitella out of it. There's still DNA of Capitella. You should never think. So that's why, for example, some people said you have to use richness. And I will tell you, don't use richness. If you use only richness, you will have species will be there everywhere, because DNA of the species will be everywhere. It doesn't mean anything. It's only because there's much more of organisms that produce the mucus. That's why this DNA is much more abundant close to the cage. So richness is not a solution. And there's a different Tubificidae or Spionidae, that are all polycats or oligocats that are present near the impact, and then other groups are not present in impact. So this relative abundance doesn't work very well, but it works somehow. So then we went to Norway. The first study was done in Scotland, and then we went to Norway, and we get this project from Norwegian Seafood Foundation with help of industry, because there was a clear interest to do this work for much more bigger scale, so we could sample all the coast of Norway with many different fish farms. If you've ever been to Norway, you certainly, when you see the farms even just flying into Bergen or any other places, there's plenty of them. It's 1,000 farms in a single country. So it's a huge environmental. This is the second economy, Norwegian economy, after oil. And it's a huge economic problem and also environmental problem, how to kind of control this. So we did a lot, up to 500, and I think now it's even more, because it's this year we also did some, so there's many samples. And well, what we wanted to do is to get out of this problem of macrofauna. Logically, if you do 5 grams of sediment, and this is upper limit, 5 to 10 grams is what maxi kit from Kayagen can do. And there's, I don't know any other kit that can do more. You could do it through manual stuff, but this is not possible for routine. You cannot use phenylchloroform stuff routinely. So you have to do this kind of kits, and they are limited. So you have to limit to the certain number, and this macrofauna-based indices doesn't work on this. So we have to get out of this macrofauna, because they're not well represented in data set, and in fact the data are dominated by meiofauna and microbiota. It's obvious, and this is what is shown here, that when you sample eDNA from a grab or core, whatever, what you mainly obtain is microeukaryotes, so protist, and meiofauna. I don't know, sometimes it doesn't work. So you see the proportion here. This is the number of sequences for one of the marker. You get this is all protists. These are meiofauna, so mainly nematodes, copepod, and platyhelminths, mussels, and this is macrofauna. So if you only work on this assign, you work on 1%, 2% maximum. When normally you work on, but the problem is that all ecological values are only for this group. There's nothing, even for meiofauna, even for few nematodes, there are the ecological values, but nothing for the rest. So most of the data are unassigned. And you have to do something about it. This is, at least we feel that when you collect the samples, okay, you don't get the sea stars or worms or crustaceans, you get some kind of forams or protists or others. So you get this, normally you get 1,000 specimens per graph in the case of macrofauna. Here you get only 5 grams, and you get about 500 or 1 million sequences from 5 grams. So we need the tools that are adapted first to this very small organism and then to this big data. And the only way we find that is possible is to use artificial intelligence instead of normal way to doing. So in this case, we use machine learning approach to analyze this data, because it's a big data problem, and you all heard about machine learning can do very much for facial recognition and any kind of problems, and this is exactly the same problem. In fact, you have a data, and you have to recognize the patterns that will tell you this site is polluted, this site is non-polluted. And you don't care about what does it mean in terms of taxonomy whatsoever. So we start to do it this way. And this is also a published paper in Science under the Science and Technology. This is for those who want to understand. You also always have to train the model. Whatever you do, you need to train the model. You cannot, machine learning will not know if you don't tell him what does it mean. So in this case, we train the model by macroinvertebrates. So we do, as I told you at the beginning, we always do in parallel, both. So we train the model on macroinvertebrates. This gives us a smiley, that gives us the ecological status. And then we do eDNA. In this case, we do on forams, so a group of protists, but then we find out that we can do it with any kind of organism, doesn't matter. And then this is trained on this kind of ecological status. You make the algorithm that will take this value, put it on these sequences, and then you can make the prediction. So theoretically, it's not nothing new. It's all what we do here. It's mainly random forest. It's something that exists in the 70s, and it has been conventionally ecologically used for many years, but had never been used to eDNA data before. So this is the only difference, but it's nothing special. And then we do the prediction using the algorithm from something that is unknown. Okay? And so this is what we also publish here. We start to do it with assigned sequences. Then we compare with assigned metazoan sequences, macrofauna plus meiofauna, and then you use all eukaryotes. And you can see how the correlation increase. Correlation increase only because you get more and more signal, because you get more and more organisms, species that are not here. Here you have very few. Here you have a little bit more because you have meiofauna, nematodes, and so on. And here you can make use of everything, of all sequences that are present in the sample. In this case, this was done on one of the marker, which is 18S V1, V2. This is the first part of the 18S gene. But then I think this is the next slide. No, this will be after. But just you can see here how this correlation increase if you do whole eukaryotes data. And from the practical point of view, what is really important for the assessment agency, governmental agencies that assess, it's how far this method is congruent with the normal morphological method. And here you can see that here you can make an error. So what is here, zero difference between, this is zero difference between morphology and molecular data, machine learning stuff. And when you do macrofauna, you have still few samples that go totally wrong. For some reason, they will not show it. And then you can see that with this is going down, and then at the end with the eukaryotes, you get majority 0 difference, and for few get 1 point difference, 1 point on the index. So it's not very much. And here there's another paper that we just published, Molecular Ecological Resources, this year, where we wanted to know whether it's depending to the marker, in fact. So we wanted to check different markers. We do it on bacteria, on forams, on eukaryotes in general, and on, this is also eukaryotes. It's all bentos, but in fact it's also eukaryotes. So V9, V1, V2. And you can check it for many different groups. It appears that it works well, the best works on these two. So somehow it works very well when you use eukaryotes as a total, as a global eukaryotic diversity. It doesn't work very well on bacteria for some reason. I don't know, we have to still continue, because I suppose that bacteria will work also good, and then you don't need to do such a big sampling maybe. But we have to do in forams, unfortunately. I like forams, I'm a specialist of forams, but it doesn't work very well. So unfortunately, it's not the best group. The best is when you use all protists or protists plus animals. So this is always this predicted. Of course, the main problem is that you have to increase the database, the calibration point, if I want. If I do it in, for example, just if we do all this work, if we get all the machine learning from Norway, and then we go to Canada, we have samples from Canada, no way. Our machine learning is unable, especially from British Columbia, where everything changed, sequence is totally different. And then we have to repeat everything again, because it will not work, because there's not the same type of sequences. It may work in Scotland or Faroes, but it will not work in Pacific, for example, if it's done on Northern Europe. So this kind of limit of geography. We don't know exactly what this is. You know, this is all pilot study, so we don't know what the limit is. There must be the limit of geography, how much the community changed. If the community changed too much, that would be different. Also, it can depend on the sediment, it can depend on the type of sediment, sandy, muddy. It may not have the same community. You have to be careful about this. There are many of these factors that we still have to, seasonality maybe as well can impact. So there may be some impacts, but generally, if you increase the size of the data, and then you can do some kind of analysis that will show you, for example, that the community that you obtain doesn't fit to others. And then you say, okay, I cannot do it. I have to find the place where I can calibrate it. So machine learning has a limit because it cannot go further than the training data set is going. That's very important. Okay, so advantages. I try to sell my methods, of course. So the advantage provides accurate predictive models, use all data set on the sequence, so there's no any limits. We use everything. We extend the range of potential bioindicators because we use the groups that nobody never thinks they can be indicators. And we can go back. Some people said now we should go back to find out what really make it. And there may be some group of proteins that are very good indicators that nobody never look at them. Of course, it doesn't require prior knowledge about species ecology, but it requires the training data set. So you don't need to know what the species mean, but you need the training data set, otherwise you cannot work. And of course, it can be automized, standardized. Of course, it's a machine learning, it's an artificial intelligence. You need just a computer to do it. So powerful computer, but nothing more. Okay, so conclusion, sedimentary DNA can be successfully used. I hope I convince you about it. This is I think it's a very, very important for the future. We have the biodiversity inventory. You still can use it to find the bioindicator species. It doesn't say that you can't use it, but you don't use all the information. And of course, if you use approach like machine learning or any other artificial intelligence, I think this is what you have to do if you have big data. Everybody understood it, but for some unknown reason, people who do high throughput sequencing accept microbiome in medical science, otherwise they don't use it. So I think this is the really good way to do it. I think that I almost finished. Yeah, I just have a few slides just to make it. this is not only, these are just 2 case studies, you know, the marine farms and diatoms. We are now working also on other application, especially for different type of industry, like desalinization plants that pollute a lot of, this is from Israel, but there's much huge industry that has a big impact on coast. Then deep sea mining that you certainly heard about, this deep sea of nodule mines in Pacific. And we have a lot of data from this, but this is all baseline study, because honestly there's no real deep sea mining yet. So it's very difficult to say, but the baseline study are very important to know what kind of communities down there before the big machines come and took all the nodules out and destroy everything. And of course the platforms is a problem. So we started by platforms because this is probably the biggest problem. We worked with Adriatic and get the project done on the North Adriatic. So this is in Italy, in near Ravenna, and get plenty of samples. This is also, you know, supported by industry. We get a lot of samples. We analyze it for all direction and all different distance from this, and we use a lot of, they wanted to have a lot of markers, so we have more than six different markers for water and for sediment as well. But at the end, when we find out, this is not, we couldn't use machine learning because we don't have a real calibration. But when we find out, the real difference was found basically at the distance less than 50 meters, which I suppose is more based on the structure of the platform, impact of the structure that the platform creates and the real kind of impact on the surrounding water. So this is one of the possibility. I think that we could continue on the oil platform, but or any kind of sites, and there are some people who do it now. We have a project with Statoil, for example, in Norway. The problem is that these platforms themselves, of course, change the environment around the platform, but until you don't have a spills, there is no real big impact on the further away. So it's not like the farms. The impact is much bigger because you put more fish into it. Here you will have to really, unfortunately, will have some kind of catastrophic, like in Gulf of Mexico, to really see the impact. Otherwise, its impact is not there. But they do it anyway on benthic, and I think we can do it on sedimentary DNA as well. This is show you because we also make this normal index, and we have seen that there is an impact at about 50 meters from, but then everything is more or less the same. So, well, I think that North Adriatic anyway is very polluted, so maybe it's not really from the farms, but from everything that is there. Okay, so just to finish with this, advantage is it's certainly faster, so where the time is important, that gives you the method that can be done with a wet lab, dry lab within 2 or 3 weeks. Probably. Sampling is much easier, so it's better to spend much less time on sampling. Then, of course, small size of samples, and it can be highly controlled in terms of sample processes, can be standardized, easy comparable data, so much less of manual, subjective fragment, and also, of course, it is based on global benthic diversity instead of just one group of organisms. So I think this basic stuff is very important, that this is global diversity. So yeah, so to finish, there's home messages, you know, we have now metabarcoding, it's called here multi-taxon community profiling, but this is basically metabarcoding with different markers. You have ultra-deep sequencing. If you add to this machine learning, you have a perfect cocktail to make a fantastic assessment of environment without anything else. But you have to combine all the things together, and then, of course, we need a lot of samples and funding to process them, because this is the main problem. It's remained relatively costly. This is a group of people that I've been working with in my lab, Frank, Philip, and Tristan, and Christina, Joanna, and Laura. These are the people who really make this work, not me. So they are really acknowledge them, and then many people who work in the field on the industry and other stuff. So this is mainly for fish farms things. Okay, and then for those who are interested, there's European Action It is called DNA Aquanet, that I am part of it, and everybody can join it. It's how to, promote this genetic impacts, because there's all, I haven't talked about it, but there's all these political aspects about regulatory, I would say, laws and rules that has to, the people, politician has to be convinced that this is the way to do. And for a moment, marine strategy or directive or water framework directive is not anyway open for the genetic work. So as long as they are not open, we cannot do anything. So if you go to DNA Aquanet website, you can see there's many groups working on barcoding, meta-barcoding, and different applications. Okay, thank you very much.
Thank you very much. So it's time for questions.
Thank you very much. Very nice talk and very interesting for several aspects, especially the practical ones on one side. So very useful method, very quick. I'm wondering whether you can then go back, because you say I will have an assessment of environmental conditions without ecological knowledge by machine learning. Can you do the opposite? Once you have got your sample sorted by quality based on machine learning, you can open the black box and see what is there and what characterizes polluted environment versus non-polluted, and so go back to knowledge, because after all, as you said, these machine learning systems are what you put in, so you're not ready for something that is not expected. So we shouldn't give up trying to learn more. And this is, and then I have a small other question. What is this? What do you think? You were surprised to find echinoderms in your samples. Big animals in a small amount of size. We have the same experience with our filters, with our work that we are doing out here, that we find even dolphins or whatever. And so what is there? What do you think it is? Because we filter on 1.2, and we can't understand whether they are cells or free DNA, really. So.
Well, just to answer the second one, because I think the answer is probably easier. I believe that all come from organelles. Some people said that this is free DNA, and it's and I think some of you already make this work on eDNA, on extracellular DNA. Some people said that extracellular DNA has been, for example, precipitated from water. It can be extracellular DNA, but then we realize that the size of the fragment is not such a big limit. People think that the size of the fragment would be a limit in extracellular DNA because the size would be, cut off. But in fact, it's not. So you can get as a huge COX-1 or even 18S, gene. So I suppose that this is just from the mitochondria. Mitochondria itself, or any kind of, if it's chloroplast or whatever you use, it's probably more like there's a paper that showed that this is in fact mostly mitochondria in the case of fishes, because they were asking where this DNA comes, that I can give you all the list of fish from just 1 liter of water. And I think it's because of mitochondria coming from mucus and others. So this is probably the answer. It's something between cells and the DNA. And for the first question, of course, we are looking through it because I'm taxonomy, so I would like to see what really makes this machine learning so successful. But it's a little bit complicated. For some groups, it's easier, but we are trying to do some kind of network analysis to figure out whether we can identify the sequence that would be attached to this particle and that are not really there. So the work is ongoing. We do practically for the meiofauna, because meiofauna it's much easier to do, and we try to, for example, disentangle these nematodes, because nematodes are everywhere and they have some kind of ecological value, but it's a nonsense because you can't give to all of them the same value. So I think we can separate them, and we can go to the species, but it will take some time. Why like that? It's faster, you know. It's like with the face, you know. You can find out what character, but also you have to understand that machine learning doesn't work on a species or sequence. It works on the combination. So it can be much more complicated, and it doesn't mean that we will find out easily one single species that is an indicator, because it's a number of species and the frequency. So it's more complicated than just single species.
So about the, if you are non-taxonomic, in some way you have to start somewhere, and you have your environment, so you choose reference environments with certain combinations of species that you say, and this is green, and this is orange, and this is red. So somehow you have to start with it. How do you do that?
So very practical, we were looking with Norwegian industry how to use it. And the conclusion was that, in fact, they don't want to use only this way, because, of course, there's a lot of opposition, because it's a black box and so on. So, you know, we still will continue. They will still continue to do morphology, but, for example, they will continue to do morphology on half of the samples, and the other half will be done genetically. Or every second year, for example, they will do morphology. Or another possibility is to do always. There's kind of you know, to be sure that this is not totally out of our hands. And I understand very well that this is a system that can very easily get out of control. You don't control this, and regulators doesn't like it. Regulators want to have control on the things, so they want to see species, not or specimen, not the sequence or black box or machine learning. This is stupid. So they want to have some kind of control. So I think that at the end, first of all, we need to calibrate. As I said, training data set. We cannot do it without taxonomists anyway, because training data set has to be done. And even we have a lot of samples, more than 500 samples, it's still not enough. There are some regions and some habitats that hasn't been samples. So what we are doing now, when we get the samples, for example, we see that the community is totally different from what we have. We said, no, we don't do it. Go back to morphology and do it morphologically. Then we can do it. Then we get the training. But this is basically. So it's a long way to get everything done. So it's not, no, in my opinion, it's not the danger for the people who are doing now. It just can increase the control easier. So yeah, I don't know whether it's also.
You can do the taxonomy and deliver these data.
Yes, yes. It's you can still be sure that don't worry. Any other questions?
Yeah, but this whole community analysis now require also sort of a reference database made out of patterns in a way, no? This analysis, because as you were saying, if you know that certain patterns are informative for a polluted site in North Europe, they might not work in the Mediterranean, or they don't work in the Atlantic. So there is also need of a different reference database, no, for the interpretation of this data. And another comment is, okay, this is one approach. However, the one you showed in the first part of your talk, no, limited to diatoms. So that's a smaller part of the community, but that's more promising, if I correctly understood. I mean, it's in a way easier and it provides good results, the analysis of the epiphytic diatoms made with the barcode.
Yeah, so if you go back to diatoms, we have been somehow criticized that in fact we calibrated with diatoms themselves. Because somehow someone could see it as a circular process. We take diatoms data from morphology, and then we use this data to calibrate sequence data and get some kind of, taxonomy-free, let's say, approach. So, yeah, not everybody like it because, and probably they're right, that this should be done on chemistry, on chemical data. as it has been done at the beginning, because at the beginning, the ecological value were given by chemical data. They were comparing the community of diatoms, for example, in Switzerland. It took them 10 years or 20 years, and they provide all the species with ecological value, and then this is published, and this is used now as bioindicators. So some people said we should go back and do chemical data again, and they do it there. But that will cost an enormous amount of money, because that means that we will have to do, we know we have the data for some places, but not everybody do chemistry anymore. So it's a problem. So I think the reference database is very important for any place you go, this kind of approach. If you have no reference database, you cannot work. That's for sure, so you have to explore it. And then at the end, you also have to somehow ask the question whether you can go to the basic data, like chemical pollution, or you can do it just by using this already existing indicators as a calibration point. That's a little bit, you know, not clear, no.
The question, otherwise we go for lunch.
Yeah,
Lunch.
I think everybody gets
No.
hungry, yeah?
Another question.
Yeah, sure.
I don't know if you said already. Did you give some attention to the things that were unclassified? Were there chimeras, recombinants? What were they?
So basically, we use a very standard procedure to clean the data. We assume there's no chimeras on them, but of course, it may be some chimeras. But I believe that because we use the whole data set, as even if there's some, we make some kind of stringent filtering. We remove everything rare, everything that is not present in all. For example, is less present than 1% in all samples or something like that. So we're not interested in this very rare kind of sequences. So I hope that things like cameras are not really big problem. But of course, you cannot really go to all the data and look through them and check everything, because this is not, the point is not to do it. So, you know, we have to assume that there may be some errors in it. Absolutely. Yes?
I have just a curiosity. Which one is the time of degradation?
Thank you for your talk. I have just a curiosity. Which one is the time of degradation of DNA in the water? I mean, how long you can, is the time of...
It has been shown that that the DNA in aquarium, for example, I think it was done on some frogs, that 2 weeks is a kind of time that the DNA will disappear. If you remove the frogs out of the aquarium, then after 2 weeks there will be no more DNA. That is a published study in 2006 already. Then there's many other studies, but we know that I think this DNA can stay a long time in the water at least. Everything this type of study never been done on sediments. It's all done from water. For all those people that use it for finding, I don't know, whales or dolphins or fish in the sea. So they consider, but the main problem is not so much, in my opinion, how long it can stay, but how far it can be transported. And it has been shown that, unfortunately, for the river system, DNA can be transported for more than 40 kilometers, which is a little bit problematic if someone wants to know where the population is. At the end, you don't know nothing, because during 2 weeks, this DNA will go everywhere. And this is what we have. For example, we also work on invasive species, tracing the fish in the rivers, and this is a big problem, because you don't know, you find the DNA, but you don't have no idea where it's come from. And this is a huge problem.
So if any of you has other questions, you can talk with Professor Pawlowski after lunch and contact Vibe, because he will lead the agenda for the afternoon.
I'm here all the afternoon, so I can discuss.
Thank you very much.